So today we will see the lecture number 2, causes of plant diseases. Today we will see what are all the pathogens which causes diseases in plants. We will see. So myself NH Shankar Reddy, doing first year PhD plant pathology in Annamalai University. So before entering into the causes of plant diseases, we will see an account on Koch postulates. So the Koch postulates are considered the fundamental aspects or the basic aspects of plant pathology to confirm a disease. So here these Koch postulates are otherwise called as pathogenicity rules. Here we have four Koch postulates which was given by Robert Koch. Yeah, out of four Koch postulates, three Koch postulates, first three, 1, 2 and 3, which was given by Robert Koch, and the fourth Koch postulates which was given by E.F. Smith. He is considered as father of plant bacteriology, E.F. Smith. So we will see one by one. The first Koch postulate is the organism must always present in every case of the disease. Like, let me tell you an example. If you take any disease, every time you are isolating an organism, the disease must be present in every case of the disease, or I mean the pathogen must be present in every case of the disease. The second Koch postulates the organism must be isolated from a host containing the disease and grown in a pure culture. So after isolation, the pathogen can be grown in a pure culture. So let me tell you an example. If you are isolating a pathogen from groundnut, for every time you are when you are isolating from the groundnut, the same pathogen must become, let me tell you, if it is a stem rot of groundnut, it was associated with the sclerotium ralphi. Every time when you are isolating the stem rot of tuberose pathogen, the sclerotium must be present in every case of the disease. That is second Koch postulates. Coming to third Koch postulates, the culture must be able to create characteristic symptoms. What are all the symptoms that sclerotium produce? The same type of symptom the pathogen also has to produce when we are inoculating into the laboratory culture. These are the first three Koch postulates which was given by Robert Koch. Coming to the fourth Koch postulates, which was given by E.F. Smith, the pathogen must be re-isolated on our comparison with the original culture. When we are isolating on the first time, we identify few of the symptoms and their few of the characteristics. When we re-isolating on the second time, it also found to be the same characters as we seen in the first. That is the fourth Koch postulate. These are the very important things that can be seen in a plant pathology. So this Koch postulates only applicable for fungi and bacteria, and the Koch postulates are not applicable for obligate pathogens. Obligate plant parasites like a virus, viroids, they can't applicable. I mean, the Koch postulates are not applicable for that type of disease. So coming to the causes of the plant diseases, here the plant diseases are mainly caused due to two types of agents, that is biotic agents and abiotic agents. So coming to the biotic means life. What are all the things that can causes diseases that comes under a biotic? The first one is fungi, second one is bacteria, virus, protozoa, phytoplasma, spiroplasma, algae, nematodes, and phanerogamic plant parasites. These are all the biotic agents which causes diseases in plants. We can see one before that, biotic agents, and the second one is abiotic agent. Abiotic agents otherwise called as inanimate agents. So the abiotic agents which causes deficiencies or excess of nutrients, which means deficiencies also causes some few of the symptoms and the yield reduction. So these are the abiotic factors also responsible for the disease losses and the quality and quantity damages. So the second abiotic factor is light, moisture, temperature, air pollution, lack of oxygen, and the toxicity of pesticides. These are all the abiotic factors which causes plant diseases. So due to biotic and abiotic causes, yield can be reduced, and also quality and quantity of the crop also can be reduced. Losses due to plant diseases, or what are the importance of plant diseases? So globally, enormous amount of crop losses can be caused due to the plant diseases. So already told you that due to the biotic and abiotic factors, the crop losses may happen. If you see, if you look into the historical evidences, a few of the diseases have a great importance in the history of plant pathology. So coming to the first one, Irish famine, which is due to late blight of potato. Actually, it is a fungal disease which is caused by Phytophthora infestans, which happens in Ireland in the year of 1845. And one more thing is the main reason for the initiation of the new branch, that is plant pathology, is mainly due to the late blight of potato, that is Irish famine. So it has a great historical importance. Due to that, nearly 2.1 million people are migrated from Europe to United States of America, and also more than 2 million people are died on those times due to the food scarcity. And the second one is a Bengal famine, which is due to Helminthosporium varese or bipolaris varese. The disease which occurs in the region of Bengal. Now the most of the parts of Bangladesh and the parts of Kolkata in those time of British times. So all the Bengal is combined. So it is that is the disease is brown leaf spot, which is caused by Helminthosporium varese or bipolaris varese in the year between 1942 to 43. And the next one is coffee rust that happens in Sri Lanka in the year of 1867. Sorry, it's not 1967, it is 1867. So these are all the epidemics which leaves a great impact on plant diseases. So why we have to study plant pathology? So coming to this, to understand the etiology or what are the reasons for this diseases, so we can understand all the reasons and etiology, environmental factors, so that we can manage the diseases betterly. So coming to the plant pathogenic organisms that I already told you, the biotic agents, coming to the protozoa, chromista, fungi, bacteria, virus, viroids, phytoplasma, spiroplasma, nematodes, phanerogamic plant parasite. These are all the biotic agents which causes plant diseases and causes huge number of losses in crop. So coming to the first one, that is protozoa. Protozoa is a eukaryote, either free living or plant parasitic plant pathogen which causes diseases in plants. So one of the protozoal diseases which can seen on plants is that is clubroot of cabbage, which is caused by Plasmodioporo brassicae. In history itself, we have seen clubroot of cabbage was discovered by Voronin. Life cycle of clubroot of cabbage is also discovered by Voronin. On the scene, we can see the diagrammatic representation. Bulging or bulking can be seen on the root regions. So this is mainly due to the production of tumor. I mean, the tumor cells due to the production of hypertrophy and hyperplasia. So due to the production of toxins and growth relating substances, it can cause diseases. So what is hypertrophy and hyper? If we come to that, hypertrophy, the abnormal enlargement of cell, is called hypertrophy. Hyperplasia, which means increase in number of cell divisions. Due to that hypertrophy and hyperplasia symptoms, the bulging or the tumors can be seen on especially root regions. Already seen in here, Oomycota comes under Kunsta. So Oomycota cell wall is made up of cellulose, but all fungi cell wall is made up of chitin. So the study of this fungi and fungi-like organisms is called mycology. Mycology is the study of fungi and fungi-like organisms. We can also call it as fungology, but the main preference will goes to mycology. So what are all the important disease comes under fungi? So wheat stem rust, Puccinia graminis, it is a great importance. So coming to this fungi, wheat stem rust, which is caused by Puccinia graminis triticale. On the right side, we can see the disease of Puccinia graminis triticale. So one by one, we can discuss in detail when we get into the subject. So it is just for a general account. So the next one is bacteria. So bacteria is a prokaryote. So far we have seen about eukaryotes. Pro means false. Karyon means nucleus. So it doesn't contains any true nucleus. So bacteria is a prokaryotic unicellular organism, whereas fungi is a multicellular organism, whereas bacteria is unicellular microscopic achlorophyllous microorganism, where their cell wall is made up of mucopeptides or peptidoglycans. Mostly bacteria can reproduce by means of binary fission. And of course, sexual reproduction will also be there, but most of the fungi, nearly 90 to 95% of the fungi, can reproduce by means of binary fission. So the coming to the morphology of a bacteria, there most of the bacteria are rod shaped. So rod shaped bacteria is called bacilli. Rod shaped bacteria is called bacilli, and the spherical shaped bacteria is called cocci. Spiral shaped bacteria is called spirali. Comma shaped bacteria is called vibrios, and thread like bacteria, that is filamentous. So I'm just giving a general account. Later on we will see in detail about one by one. So the study of bacteria is called bacteriology or plant bacteriology. So on the right side we can see the bacteria diagram. So flagella can be seen on bacteria, only pili can be seen, DNA can be seen. So different types of structures also there in bacteria that we will see one by one. On the right side down, this is the first plant bacterial disease to be discovered, that is fire blight of apple, which is caused by Yersinia amylover. It is a plant bacterial disease and first plant pathogenic bacterial disease to be discovered by T.J. Burrell. In the history itself we have seen that is the first plant bacterial disease which was discovered by T.J. Burrell is fire blight of apple. Why it is named as fire blight of apple? If we see that fire blight infected plants from the long distance, the firing symptoms can be seen. That's why it is called as fire blight of apple, which is caused by Yersinia amylover. But virus contains RNA, either RNA nor DNA, but not both. Any one type of genetic material is present in the virus. But viroids only contains RNA as a genetic material. So the coming to the virusides. Virusides are circular single-stranded RNA-dependent plant viruses. For replication and encapsulation, we can see the different types of tiny structures on the diagrammatic representation. Viroids, prions, virusoids, virion, virus, different types of structures already there. So the next one is a Phytoplasma and spiroplasma. So Phytoplasma are wall-less. It contains no cell wall, bilayered, round, elongate, prokaryotic organism. So Phytoplasma doesn't contains any cell wall. It is a bilayered structure. The Phytoplasma contains both the DNA and RNA as genetic material. So it mostly belongs to the order Achiloplasmatales and belongs to the order Phytoplasma. So best example for Phytoplasmal disease is a little leaf of brinjal. Next one is the spiroplasma. The spiroplasma is also cell wall-less helical motile prokaryotic organism. It is a motile prokaryotic organism which contains a triple layer membrane. So Phytoplasma contains a double layer, that is bilayered member, whereas a spiroplasma contains a triple layered membrane. So spiroplasma belongs to the order entomoplasmatales and belongs to the genus Spiroplasma. The best example is constant. So on the right side we can see the diagrammatic represent. On the left is that is Phytoplasma. On the right side is the helical like structure or thread like structure, that is Pyroplasma. So next one is the nematodes. So nematodes also be the responsible for the causing of plant diseases. So normally nematodes are worm-like structure which causes a plant disease and produce galls and tumors in plants. On the right side we can see the diagrammatic representation which is caused due to nematodes. One of the important nematodes is root knot nematode, that is caused by Melodogeny. So the first nematode to be discovered is Anguna tritici by Needham. So it is the Anguna tritici is the first nematode to be discovered. So the next one is phanerogamic plant parasites. So phanerogamic plant parasites are the parasites which causes plant diseases and absorb the nutrients, so that due to the absorption of nutrients from the plant cells it causes losses to the plants. So it dependent on another vascular plants for food and water. So few of the examples is Cuscuta, Loranthus, Vorabanki, Striga. On the left side first picture, that is a thread like yellow color thread like structure, that is Cuscuta. So it covers all the plants so that it will be very difficult for the plant to respirate, and moreover it absorbs the nutrients from the plant cells. So the second one is Loranthus. We already seen the history itself. Loranthus is the first plant parasite to be discovered by Albertus Magnus in the 12th century. So it has a different names, that is called giant mosquito or mosquito dendroptoi. So Loranthus has a different names. In the second picture, that is Loranthus. That is Loranthus. The third one is Vorabanki. Vorabanki. The third picture is Vorabanki. We can see in all the fields of maize, on the fields of sorghum, the fields of the different fields have been seen. And the fourth one is Striga. The fourth one is Striga. Pink color flowers can be produced by Striga, that is Striga. So this all plant phanerogamic parasites can absorb nutrient through hostoria. So these all plant phanerogamic parasites produce hostoria so that nutrients can be absorbed through hostoria. So what is hostoria and all those things we can see in the general character, so that it will be very easy to understand all the concepts. Okay.